Hello and welcome everyone. I'm Sumit Vohra, working as a researcher at Zeus Institute Berlin, and our work is very much at the intersection of the visual data analysis and neuroscience. Today, I would like to present the Hypothesis Explorer, which is an analysis tool that originated from our collaboration with the Engert lab at the Harvard University. Let's start with the definition of the neural circuit. It's a collection of neurons connected by the synapses and together perform a specific functions when activated. On the right hand side, you see one of the Cahl's drawing, which shows the contralateral organization of forebrain. In modern terms, Cahl drawing would be called a neural circuit. These neuron pathways can be dedicated or distributed. For example, there is a direct pathway from the sensory input to the motor nerve cell. Or there is an indirect pathway which first sends the signals from the sensory input to the spiral fiber neuron. Then the spiral fiber neuron sends the data to the motor nerve cell, which then led to generate the escape response with the help of the motor neurons. Next, let's take a look at the model organism we are dealing with here. There are several animal models in study. For example, monkey, rat, zebrafish, and medaka. We work with the larval zebrafish here because they are transparent in very few days and whole brain imaging is possible without any surgery. Other advantages are there are around 100,000 neurons, breed fast, cost little, and have genes that are easy to manipulate. On the right hand side, we show a video of a larval zebrafish performing about swimming. Next, we tested our tool on two real-world case studies. First shows the optomotor reflex, which causes many animals to orient toward a visual motion stimuli and is thought to be important for compensating the involuntary drift in the flowing streams. In this behavior experiment, animal was presented with a coherent dot motion of approximately 1,000 dots projected from below in a circular arena. These dots are presented at four different coherent levels: 0%, 25%, 50%, and the 100%, either moving leftward or rightward relative to the body orientation of the fish. The second case study utilized the data from experiment designed to find network elements that inhibit escape to the non-threatening stimuli, that is the dimming stimuli. What is a dimming stimuli? An object approaching on a collision course was simulated by a dimming disk on a light background and presented to the animal from the side, while the behavior of the animal was monitored from the top. Next, let's take a look at the zebrafish data we are dealing with here. We work with 320 anatomical regions dividing into non-overlapping and overlapping brain regions. We also have the 3,000 neuron morphologies reconstructed using the stochastic techniques. Using this 3,000 morphologies, we also generated two synthetic data set of 25,000 and the 50,000 neurons each, which are utilized in evaluating the performance of our tool. Next, discuss the biological workflow. Overall, there are 6 main steps for covering neural circuit, but our focus is on the step 5, that is the exploration of the hypothetical neural circuit. Still, I would like to present the other portions of the workflow. First, a behavior experiment is performed and the activity of the neural cells is recorded using the calcium imaging in the multiple Z-planes. Next, the Z-planes are registered to the reference brain. The bodies of the active neurons are segmented and functionally characterized based on their activity curves. Next, from the large set of the reconstructed neurons, those spatially close to the active cells are determined. Based on the available information and the pre-existing hypothesis about the neural circuit, data-compatible circuits are constructed and explored using our tool. The constructed neural circuits can further be tested using ablation experiment. These goals is reflected in the following task and the tool components. Here I will explain each task and their mapping on the tool components. Each component will be discussed in detail in the subsequent slides. The region finder, as shown in the figure A, can be used to identify brain regions of interest in terms of their functional information. The component consists of the several subcomponents, including a hierarchical tree map and the bar chart views. The hierarchical tree map provides an overview of the spatial distribution of how the neural population are spatially distributed across brain regions. Next comes the query builder, as shown in the left portion of the figure B, which allow user to identify neuron subsets. The generated circuit can then be visualized using the circuit viewer, as shown in center portion of the figure B. Next comes the pathway browser, which allow users to explore all the possible pathways related to the current query, as shown in the right portion of the figure B. To enable user to perform the connectivity analysis, all the neurons contained in the circuit are then passed to the neuron browser. Finally, the selected neurons and the anatomical context are visualized in an anatomical viewer, as shown in the figure D. Let's discuss about the query builder component. It allows the user to extract subset of neurons with certain attributes from a neuron database. Technically speaking, it enables the user to create complex database query interactively by simply dragging the items and selecting the operators. It consists of two juxtaposed trees. The left tree contains all the components that are needed to build a circuit query, for example, the operators, predicate, and the entities. The right subtree is a representation of the nested filter query, which produces a filtered set of neurons that realize the neural circuit. Next component is a pathway browser. It lists all the possible pathways related to the current query. I will provide you a detailed example of how these corresponding pathways are generated in the subsequent slides. If a set operation is selected, the pathways will be enclosed by an envelope. The next component is the circuit viewer. It provides an intuitive visualization of the built neural circuit and the corresponding pathways. It's based on the real anatomy of a zebrafish. The connections between the nodes are drawn using the smooth curves. An arrow at the end of the curve indicates the terminate in relationship. Two locations per brain region are shown to distinguish between the left and the right hemisphere. The subqueries in the query tree can be collapsed or expanded as needed. If the set operation is collapsed, the pathways will also be collapsed in the pathway browser. Aggregated regions in the pathway will be shown as a colored pie in the pathway browser, preserving the location of the left and the right hemisphere. When users select an aggregated Note, by default, the links to all the corresponding regions sets are shown. However, it can also be displayed as an aggregated edge. For example, the red line leading to the orange region as shown in the figure. Next, we will discuss how query operation led to the neural pathways, and we will illustrate this using a simple query operation. The exact query here is to filter subsets of neurons having their somas in the forebrain, transit through the midbrain, and terminate in hindbrain. The query was performed using a simulated dataset with around 50,000 neurons. We start by dragging the intersect operator and the forebrain region into the filter query component. And next we drag the filter predicate soma in and apply it to the forebrain. By default, it shows the number of neurons for both the left and the right hemisphere. Next, we drag the midbrain and apply the filter predicate transit into the midbrain. The pathway browser shows all the 4 possible combinations sorted by the number of neurons. These connections are from the forebrain left hemisphere to the midbrain right hemisphere, and for the forebrain right hemisphere to the midbrain left hemisphere, and so on. Finally, we drag the hindbrain into the query component and apply the filter predicate terminates in. The pathway browser now shows the 8 possible connection between this. And next, let's build the real-world circuit using our tool. And our task here is to find the network elements that inhibit escape to the non-threatening dimming stimuli. We already explained the dimming stimuli in the previous slides. As shown in the figure, there is a circuit hypothesis that there are neurons that goes from the optical tectum directly to the glycinergic neurons, which in turn connect to the Mothner cells that will finally generate the escape response. The optical tectum can be further divided into 6 tactile layers, as example SO12, SFGS12, etc. Then using the query builder, the expert try to find such a neurons that pass through these tactile layers and having their somas in the midbrain, and then they transit through the tectum and they have the terminates in the caudal hindbrain. Experts were able to find 5 such neurons as shown in the anatomical viewer. The circuit viewer shows the built neural circuit. Next, in order to obtain the qualitative feedback and further improve the tool, we conducted a user study. We invited participants from the field of neuroscience, computational neuroscience, and visualization. In total, 16 users participated in the survey, of whom 9 were the data scientists or visual researchers, and the 7 were potential users from the neuroscience. We asked them several questions like whether the query functionality is easy to use and the components provided in the panels are appropriate. The pathway browser make it easier to explore pathways and filter neurons of interest. Next, we asked participants to rate these questions using the 5-point Likert scale. On the right hand side, one can see the results of the user survey. The first 4 questions were rated positive by all the participants. The response for the question 5 was mixed. The tool was rated positively by the neurobiologists, but the data scientists were not sure how it could help them in their research. In summary, we presented a visual analysis tool that allows neuroscientists to quickly extract those pieces of information that are relevant to their research questions. We showed the applicability of our tool on the 2 real-world case studies. As a biological contribution, we helped neuroscientists to narrow down hypotheses about circuit models. Given different ontological tree and variants of anatomic reconstruction, we will allow neuroscientists to compare hypothetical neural circuit in future. Uh, I would like to acknowledge my project partners from the Harvard University. This concludes my talk, and I would be happy to answer your questions.